So you'll want to open up the website that I have here so that you can follow along as well, because there's some activities that you can do and check your work. I'll include a link to this on Edline as well, or you can just type the address into your browser. So in the Bean Lab, you discovered that natural selection isn't the only way that evolution can happen. If evolution is not happening, we say that a population is in genetic equilibrium. And Hardy and Weinberg were two scientists who came up with 5 different conditions that must be true of a population if they are not evolving. So let's go through those. Conditions. The first one is a large breeding population. Okay, so if there's a large breeding population, small random events aren't going to affect it as much as if there's a small population. So for example, here we've got a map that shows a population that lives in California. It shows a group of people that live in California where there's 10 million people in 1997. And all of these people in California could potentially mate with each other. However, there's a natural disaster and 3 of those people in California die. Well, taking their genes out of the gene pool when you have a total population of 10 million isn't going to have a very big effect. However, if you have only a very small population, like we did before the gold rush, then killing just 3 people is going to make a big difference in the gene pool. So the smaller a population is, the more likely a random event is going to affect their. These random events are called genetic drift. The second condition that's required for genetic equilibrium is random mating. This isn't something that we simulated in our Bean Lab because I think it was pretty difficult to simulate. But random mating means that individuals don't choose their own mates. So an example of random mating is the way that corals mate. It's actually the way that a lot of ocean animals mate, is they just release their gametes into the water. And they hopefully will randomly meet up with a gamete that is compatible with theirs, so of the opposite gender and of the same species. More likely with animals that live on land is choosing their own mates, and so sometimes this is called assortative mating or non-random mating, where individuals mate with other individuals that look like them. So here we've got 2 large bugs that mate and 2 small bugs that mate. A third condition for equilibrium is that there's no mutations. Mutations can affect the allele frequency because here we've got 100% light brown alleles when there's no mutations. And now if we introduce a new allele into the population, now the allele frequency of this light brown or tan allele goes to 75%. So this change in allele frequency is again evolution. No immigration or emigration means that no new alleles come into the population and no alleles are able to leave the population. And finally, no natural selection. So no phenotype can provide an advantage in terms of fitness. All phenotypes in the population have to provide an equal amount of fitness. Okay, so Hardy and Weinberg came up with an equation that we can use to determine if a population is evolving or not. And also a way to determine whether, or the allele frequencies within a population, if we know that population is not evolving. So looking at this population of pigs, the population of pigs has 2 different colors. There's black pigs and there's pink pigs, and 2 alleles are controlling this trait. So we can predict the frequency of the alleles if we know that the population is in equilibrium. Just looking at the population, knowing nothing else about it, can we count up the number of recessive alleles? Well, no, we can't. We can count up the number of recessive individuals, but we don't know of the individuals with the dominant phenotype which ones are heterozygous and which ones are homozygous. But if we know that the population is in Hardy-Weinberg equilibrium, that we've met the 5 conditions that we just discussed, then we can figure out what the frequency of the alleles are. So there's 2 different equations that use some of the same variables, which allow us to substitute in. So P is the frequency of the dominant allele, and Q is the frequency of the recessive allele. We know that there's only 2 options, so P plus Q must equal 1. So in other words, the sum of those allele frequencies has to be 100%. In this case, in the Hardy-Weinberg equation, we use decimals instead of percentages, and so that's what the 1 is, and not the 100%. So if we look at the second part, P squared plus 2PQ plus Q squared equals 1. So what that means, essentially, is, so P squared is the chances of having a homozygous dominant individual. So the chances of 2 big A alleles being together. 2PQ is the frequency of heterozygous individuals. So there's 2 ways that you can get heterozygous individuals. You could have a big A and a little a, or a little a and a big A. And I know those sound exactly the same because they are, but there's 2 different ways to get those. So heterozygous individuals are 2 times the big A frequency times the little a frequency. And then the homozygous recessive, those are the Q squared individuals. Let's see if I can reset this quickly here. No, it's not going to let me. Okay, so when you're asked to calculate Q squared, what you need to do is count up the individuals that are homozygous recessive. So in this case, black coats are recessive, so it tells us that right here. The allele for black coats is recessive. So we've got 1, 2, 3, 4 black pigs. So 4 out of the 16 individuals are recessive, and so Q squared is equal to 25%, or if we're putting that in decimal form, which we always need to do with this equation, then the decimal form is 0.25. Okay, so the next thing we want to do is find Q. Well, in the question right before, we figured out that Q squared, the percentage of recessive individuals, was 25%. So Q squared is equal to 0.25. And now if we just want to find Q, we can take the square root of Q squared to find the allele frequency of the recessive allele. So the square root of 0.25, if you put that into your calculator, should be 0.5. And if I check my answer, okay, same thing, Q equals 0.5. So now that we know the allele frequency of the Q allele, or the recessive allele, now I can find the frequency of the dominant allele P, because I know that there's only 2 options in terms of alleles. There's the capital A and there's the lowercase a, or in this case the black or the pink allele. So if 0.5 or half of my alleles are recessive, then I know that half of my alleles are dominant. So I can plug that into the equation P plus Q equals 1. So 0.5 plus Q, or plus P, equals 1. So I can just use algebra to solve that. The frequency of the P allele should be 0.5. And let's scroll down. Oops. Okay, and then finally we want to know how many heterozygous individuals there are, or 2PQ. So we've solved for Q in number 2, and we've solved for P in number 3. So we just need to plug that into the equation 2PQ. So 2 times 0.5 times 0.5. And so this should give us the percentage of the population that's heterozygous. So that should also be 0.5. And if I check my answer, 2 times 0.5 times 0.5 is 50%. So 50% of my population up here is heterozygous. We said that 25% of the population, or 4 out of 16, were homozygous recessive. 50% of the population is heterozygous, and so then just by default, we would know that 25% of the population is homozygous dominant. We would be able to calculate that by calculating p squared. So p, let's see, here's where we found p in number 3. So p was 0.5, p squared, or 0.5 squared, is 0.25, or 25%. So 4 of these pigs, or 25% of these pigs, are homozygous dominant. So let's try another example. Here we've got a population of 1,000 fruit flies. Some have red eyes and some have sepia eyes. So 640 flies have red eyes, and sepia, it tells us, is the recessive trait. So how many individuals would you expect to be homozygous for the red eye color trait? So in all of these problems, the very first step is always to calculate Q squared. In other words, what is the percentage of homozygous recessive individuals? Well, in this case, you're told the percentage of homozygous dominant individuals. Or excuse me, you're told the number of individuals with the dominant phenotype. And so just by default, you can subtract to find the number of individuals with sepia eyes. So 1,000 minus 640 is 360 individuals that have sepia eyes. And we know their genotype must be small q, I guess, for lack of a letter that they've given us. So we want to start by calculating what is Q squared, or what's the percentage of individuals with sepia eyes. So we said 360 individuals out of 1,000 have sepia eyes, so 36% have sepia eyes. So Q squared for this population is 0.36. The second step is always to find the allele frequency of that recessive allele. So in order to find the allele frequency, we can take the square root of 0.36 and we get 0.6. So now we know that 60% of the alleles in the population are sepia, and to have a total of 100%, that means that 40% would have to be the dominant allele for red. The question asks us to determine the number of, homozygous dominant individuals, or homozygous for red eyes. So the part of the Hardy-Weinberg equation for homozygous dominant is p squared. So we just found that p was 0.4. So if we square 0.4, that gives us the percentage of individuals that are homozygous red. So 0.4 squared is 0.16. And that means that 16% of this population is going to be homozygous dominant. And in a population of 1,000, that means 160 total individuals will be homozygous for that red allele. There's a couple of other example problems on here that you can try, and we're going to spend some time in class working on Hardy-Weinberg problems as well.